So, as far as learning objectives, we are going to discuss acid-base balance and the pH scale. We're going to discuss the properties of water and define solvent, solute, solution, hypertonic, hypotonic, isotonic, hydrophilic, and hydrophobic. We're going to describe the common properties of all organic molecules. We're going to name the monomers of carbohydrates and describe the structure and function of disaccharides and polysaccharides. We are going to describe the structure and functions of amino acids, peptides, and proteins, name and describe the structures and functions of the different lipids, and then describe the structures of nucleic acids and nucleotides. Name and discuss the function of the different nucleic acids and explain complementary base pairing. So when we talk about inorganic compounds, we are talking about molecules that do not contain carbon. Now, there are a couple of exceptions to inorganic compounds, and they are classified as inorganic compounds even though they do contain carbon. So a couple examples are CO2 or carbon dioxide and CO or carbon monoxide. Okay, when we talk about acids and bases, so really important to understand that the strength of acids and bases is determined by the hydrogen ion concentration of the water in which they dissociate. So the higher the hydrogen ion concentration in the solution, the more acidic the solution is. And a low hydrogen ion concentration of a solution indicates a basic solution. So, acids release hydrogen ions, and bases are substances that release hydroxyl ions. Okay, the pH scale. So the pH scale measures the alkalinity or acidity of a solution. The pH scale is based on the hydrogen ion concentration. It runs from 0 to 14. Pure water, pure H2O, is absolutely neutral with a pH of 7. If something has a pH of less than 7, it's considered an acid, and if it has a pH greater than 7, it's considered a base. So the higher the hydrogen ion concentration of a solution, the lower the number is on the pH scale, and the more acid the solution is. The higher the number on the scale, the higher the hydroxyl ion concentration, and the more basic or alkaline the solution is. Okay, so this is a figure that's in your book. It's figure 2.9, and you'll see here that there's an increasing hydrogen ion concentration. With increasing hydrogen ion concentrations, the solution becomes more acidic and the pH value decreases. When the hydrogen ion concentration decreases, the solution becomes more basic and the pH value increases. Another thing I want you guys to notice about this pH scale and the examples that are on it is that you see that most of the fungi, bacteria, and protozoans actually live somewhere in that neutral scale or that neutral section of the pH scale. You don't see very many bacteria or fungi or protozoans that exist in a too alkaline of an environment or too acidic of an environment. Okay, when we talk about buffers or biological buffers, it's important to know that they can be both intracellular or extracellular, and they're very heavily buffered, all biological fluids. So buffers are chemicals that can absorb hydrogen or hydroxyl ions. They can resist pH changes of a solution, and they're essential for maintaining the pH necessary for the survival of living cells. Okay, salts. These are substances that dissociate in water and do not release hydrogen or hydroxyl ions. They're formed by acid-base reaction or metal-acid reaction. Different salts can actually stimulate sensations of all 5 basic tastes. Salty is sodium chloride, sweet is lead diacetate, sour is potassium bitartrate, bitter is magnesium sulfate, and umami is monosodium gluconate. So we talk about the specific naming of salts. It starts with the name of the cation, and it's followed by the name of the anion. If we take a look here, we can see common salt-forming anions and cations that are shown here. So you see the anions on the left, and then you see the cations on the right. Okay, and now we get to our favorite, water. So water is the most abundant molecule in the human body and in microorganisms, which contain at least 70% water, and the Earth's surface is 71% covered by water as well. So it's necessary for all life forms on Earth. It exists in 3 different temperature-dependent states. Think about it, solid, liquid, and gas. It's a very important solvent. It's held together by hydrogen bonds. It consists of hydrogen and oxygen in a ratio of 2 to 1, so H2O, and it's a polar molecule. If you look here at this picture, you'll see that water actually acts as the solvent. Sodium and chloride ions are the solute. And when solutes are dissolved or dissociated in a solvent, a solution is formed. Okay, so then we talk about different water solutions. You can have an isotonic solution. This is when the solute concentration equal on the inside and the outside of the cell. Then you can have hypertonic, where the solute concentration in the cell is less than outside of the cell, and hypotonic, where the solute concentration in the cell is higher than outside of the cell. So let's talk about this a little bit more. Maybe this will help for this to make sense. So cells placed in an isotonic environment will remain unchanged. If you put a cell into a hypertonic environment, that cell will lose water and shrink. If you put a cell into a hypotonic environment, it will take up water, swell, and eventually burst. If you take a look here, this kind of illustrates this. So remember, cells in an isotonic environment will remain unchanged. So you see that's the very bottom row, that isotonic environment, before and after, and it looks the same. Then when you look at hypertonic, that's the middle row. You see that cells placed in this hypertonic environment will lose water and shrink. So you see it shrinking there in that after picture. And then in a hypotonic environment, that water moves into the cell and essentially expands the cell, and eventually it will burst. Okay, because of their polarity, ions attract the polar water molecules that surround the ions, which in turn attract other water molecules to form hydration spheres around each ion. Formations of hydration spheres are responsible for the solubility of ions in water. So when we talk about hydration spheres again, this is when ions attract polar water molecules. A hydrophilic means it's water soluble, or they're water loving compounds, and hydrophobic means that it's insoluble in water or it's water repelling. Okay, and if you look here, you'll see that these are water molecules from hydration spheres around ions. Okay, when we talk about organic molecules, Remember, all organic molecules contain atoms of carbon and hydrogen. The major organic molecules in living organisms are the carbohydrates, proteins, lipids, and nucleic acids. Each of these compounds is composed of a specific unit of molecules or monomers. Organic molecules have a backbone of chains or rings formed by the carbon and hydrogen atoms, referred to as the hydrocarbon backbone. So here you can see figure A is chains, figure B is the rings, and then figure C is the backbone. Okay, so now I'm sure that carbohydrates are not, this is not new terminology for you, just like probably a lot of the concepts in this chapter have not been new. It's simply a review. But when we talk about carbohydrate, carbohydrate means hydrates water of carbon. It's derived from a 2:1:2 ratio. So it contains 2 of carbon, 1 hydrogen, and 2 oxygen. Monosaccharides are simple sugars that contain 3 to 7 carbon atoms in an aldehyde group or a keto group. You see examples of monosaccharides there are glucose, fructose, galactose, ribose, and deoxyribose. Now, disaccharides are compounds formed when 2 monosaccharides combine with the loss of a water molecule. So you see sucrose is a combination of glucose and fructose, lactose is a combination of glucose and galactose, and maltose is glucose and more glucose. Polysaccharides are formed when many monosaccharides combine to form a large compound. Starch in plants and glycogen in animals are polysaccharide storage forms of glucose. The most abundant polysaccharide is cellulose, a major component of the cell walls of plants, fungi, and most algae. If we look here at figure 2.14, you see the carbohydrate structure of a disaccharide and a polysaccharide. So the disaccharide's there on the left, and then you see the polysaccharide on the right. Okay, proteins. So 2 amino acids joined together form a dipeptide. 3 amino acids form together for a tripeptides, and a chain of 10 or more amino acids form a polypeptides. So, proteins, also known as amino acids, there's 20 naturally occurring amino acids, all consist of an amino group, a carboxyl group, and a variable side chain. The dipeptide, again, is 2 amino acids joined by a peptide bond, and a polypeptide is 10 or more amino acids joined together. For those of you who are visual learners, you will like this slide. Here are some different examples of naturally occurring amino acids. Some of them you may recognize, some of them you may not. Okay, structural arrangements of proteins. So proteins occur in 4 different structural arrangements. Okay, the first one is a primary structure. This is just a single chain of amino acids. So examples of a primary structure protein are small hypothalamic hormones such as the gonadotropin releasing hormone and thyrotropin releasing hormone. So that's GRH and TRH. Gonadotropin releasing hormone is GRH and the thyrotropin releasing hormone is TRH. Those are examples of primary structure proteins. Then we have secondary structure proteins. These have a helix or a B sheet. So, examples of helical proteins of secondary structures are keratin, myosin, and collagen, and silk represents a B-pleated sheet. Then you have tertiary structure proteins. Examples of tertiary structure proteins are enzymes and some peptide hormones, such as insulin. And then you have a quaternary structure, and an example of a quaternary structure protein is the hemoglobin molecule. And so here's some different structural arrangements of the proteins. You'll see the primary structure, the secondary structure, the tertiary structure, and the quaternary structure there, for those of you who like visuals. Okay, lipids. So lipids are also known as triglycerides. They're fats and oils. They can be saturated or unsaturated. They consist of glycerol and fatty acid chains. They're insoluble in water. And they take on different forms depending on the room temperature. So at room temperature, a fat is solid and oil is liquid. Okay, so here the triglyceride molecule consists of glycerol and fatty acid side chains, as shown in figure 2.17 on the left. On the right, figure 2.18, it reflects saturated fat with no double bonds, with the carbon chain and polyunsaturated fat with several double bonds within the carbon chain. Okay, cell membranes are composed of a phospholipid bilayer with the tails facing towards each other and the heads facing outward, making their surfaces hydrophilic and their interior hydrophobic. So, you'll see the phospholipid bilayer is seen in plasma membranes there on the left. And then on the right, you'll see the phospholipid molecule is composed of a polar hydrophilic head and a nonpolar hydrophobic tail. Okay, we're still talking about lipids, guys. So, steroids have a carbon skeleton consisting of 4 fused rings with various functional groups attached. Hundreds of different steroids have been found in plants, animal, and fungi. Anabolic steroids interact with androgen receptors to increase muscle and bone mass. So when you hear about gymnasts or athletes taking steroids, it's typically anabolic steroids that they're taking. Then there's sex steroids. They're responsible for the secondary sex characteristics of males and females. Then you have mineral corticoids. Mineral corticoids help maintain blood volume, electrolyte balance, and osmolarity by controlling the renal secretion of electrolytes. You have glucocorticoids. These are often prescribed to reduce inflammatory conditions. One common glucocorticosteroid is prednisone, and I'm sure most of you have heard of prednisone and or taken prednisone and or administered prednisone throughout your nursing career. Another type of steroid is a phytosterol. Plants contain a wide range of phytosterols that are a structural component in their cell membrane and serve the same function as cholesterol does in animal cells. So if you look here, you see various steroid models that are shown, all with 4 fused carbon rings, but differing in their functional groups. So you see cholesterol, estrogen, and testosterone. Okay, cholesterol. Cholesterol plays a major role in many biochemical processes, such as in the metabolism of fat-soluble vitamins, and acts as a precursor for vitamin D. Cholesterol is also a precursor for the synthesis of steroid hormones and is important component of cell membranes. Prostaglandins play a major role in a variety of body and cell regulatory processes, including involvement in defense mechanisms like blood clotting and inflammation. Nucleic acids. So main functions of nucleic acids include storage of genetic information or DNA, directing protein synthesis or RNA, and energy transfers. DNA is responsible for inherited characteristics, growth, and cell reproduction. Okay, so the bases in a nucleic acid polymer can form hydrogen bonds with the neighboring bases by a process called complementary base pairing. 5 possible nitrogen bases are subdivided into 2 main groups. You have purines with adenine and guanine that have a distinctive 2-ring structure, and then you have pyrimidines with cytosine, thiamine, and uracil with a single ring structure. So here you see a DNA molecule. Deoxyribonucleic acid, or DNA, is composed of nucleotides, each of which consists of a phosphate, a pentose sugar, and a nitrogen base. The nitrogen base of nucleic acids are either A, purines, or B, pyrimidines. The pyrimidine uracil is only found in RNA. If you guys have any questions, please feel free to set up office hours with me. Typically, remember, my office hours are on Friday from 10:30 to noon. If that does not work for you, please try to email me and we can arrange a time to have office hours elsewhere.